Okay. So once we have our fascicle, and again, it has its own connective tissue membrane, we can combine several fascicles. So again, if we zoom out, we get the same kind of image. And I always think that it looks like spaghetti noodles. So I'm sorry, I keep using the same analogy. But again, if you group a bunch of these fascicles together, that's the actual muscle itself. And that has its own connective tissue membrane there, the epimysium. So I really like this figure because, again, you can look at it starting from the most zoomed out and looking at the muscle, and then going down to the fascicle and then to the muscle fiber, or in the order that I described it. But you should be aware of this figure here and the structures here. So you should be comfortable with a bunch of myofibrils come together to form the muscle fiber, a bunch of muscle fibers come together to form the fascicle, and then a bunch of fascicles come together to form the muscle. All right, so as far as glossary terms, sarcolemma, like I said, is the fancy name for cell membrane. Sarcoplasm is the fancy name for cytoplasm. But where we're really going to get into is when we talk about sarcomeres. So this structure of myofibrils with the Z discs and the H zone, I'm going to tell you right now, I don't care for this as much. I prefer to look at sarcomeres with this figure down here at the bottom. All right, so this image here with the H zone and the Z disc, I wouldn't ask you necessarily what a Z disc is, but I would be more interested in the function of the sarcomere and the cross-bridge cyclings that we're going to get into in a second. Because again, we're looking at the function of muscles and how they are generating that contractibility. So when we look at the structure of the sarcomere, again, we see really this almost repetitive nature, this sort of layering nature that we saw in the previous slides. But if we look at the sarcomere, come on, pointer, we can see there are these layers of fibers here. So we've got the blue actin fibers and we've got the red myosin fibers. And it's layer upon layer upon layer. Now the one part that I do want to point out is this M line here in the middle of the sarcomere. Okay. So that M line here is unique in that it is where there is only the thick filaments, the myosin filaments. There are no actin filaments in this center region here. Right. In addition, we do have these yellow guys here, and these are elastic filaments. And what they do is they help with recovery of the muscle. So we have the sarcomere during contraction, the sarcomere itself shrinks down, and then when we're done contracting, those elastic filaments help return the sarcomere to the normal size, the normal length. All right.
Sorry, can you repeat the M line function?
So the M line, I didn't talk about the function. I just talked about the fact that it is unique, or it's a noteworthy area because this is where we don't have any actin. The only fibers that we have are those myosin or thick fibers. Are there other questions on sarcomere? So we've got these thin and thick filaments, the actin and the myosin, and this is where we're going to focus on for the next several slides. Right. So on this slide here, we're looking at the myosin, and again, these are protein filaments. And the part of the myosin filament that we're really going to focus in on is these globular head units here, because these globular head units here are what are going to interact with the actin. So if we look at just a single myosin filament here, we see these heads, and these heads are where all the magic is happening. We've got a couple spots here that are going to be binding to those actin filaments. And then we've got another spot right down here that's important, and that's for the binding site of ATP. If we look at the actin filaments a little bit more closely here, I just want to point out the fact that the actin filaments here also happen to have a binding site, and they have binding sites for myosin, and that makes sense because we know myosin and actin are going to bind together to generate the contraction. But it also has binding sites for a couple other things. The two that I've put here in bold are tropomyosin and troponin. So troponin itself is a regulatory protein that helps regulate or helps bind tropomyosin to actin, and it also will bind calcium. And I'm bringing that up because we didn't talk too much last week about calcium, so I want to make sure I talk about it today. So if we look at the triad, we did cover this a little bit last week, but we know that we are looking at individual sarcomeres here, right? But we know that surrounding the muscle fiber or the muscle cell, we have a cell membrane, and that is shown right here. So that is the sarcolemma. The other thing that I want to point out to you is the sarcoplasmic reticulum. And the sarcoplasmic reticulum is just the fancy name for smooth ER, or smooth endoplasmic reticulum. So this is an organelle that we learned about, in Bio 101, the smooth ER. But because it's in the muscle, we give it a fancy name, sarcoplasmic reticulum. The important part about the SR is that it is responsible for regulating calcium levels in the muscle cell. And again, I want to make sure I highlight the role of calcium, because I do think that I accidentally didn't discuss it very much last week. All right, so the triad is essentially the connection or the junction between the cell membrane, the sarcolemma, and the sarcoplasmic reticulums. So the sarcolemma, now remember, is just a fancy name for plasma membrane. Now the plasma membrane or cell membrane actually juts into the interior of the cell a little bit. So we're used to seeing cell membranes as just sort of a circle around the cell. But in muscle cells, that cell membrane actually will protrude into the cell a little bit. And those protrusions have a fancy name, T-tubules. So I just don't want you guys to get confused with all these new terms, because they are just fancy names for structures that we've already learned about in cells. Right. So we did look at the contraction of muscle fibers last week. Again, we looked at sarcomeres. That is the sort of functional unit of our muscles. And we can see here we've got the sarcomere as we saw it on the previous slide. And again, here is the M line. So we know that we have these layerings of the actin and the myosin filaments, actin, myosin, etc. And when contraction occurs, the actin on the left-hand side is going to move towards the right, towards the M line, and the actin filaments on the right-hand side are going to move towards the left, again, towards that M line. So this happens simultaneously so that we get that sarcomere shrinking down, getting shorter. And that's because, again, those actin filaments are moving towards the center of the sarcomere in the region called the M line. So when the muscles, or when the sarcomere is fully contracted, it would look like this bottom image. And now we see why we've got the M line here. So we've got the M line that was previously only the myosin filaments present. But if you notice here, when we are fully contracted, the actin filaments that were on the left-hand side and the actin filaments that are on the right-hand side are essentially meeting in the middle of the M line. So the question earlier about the function or the importance of the M line, that would be the importance of the M line, that is the center of the sarcomere, and during full contraction, that is the point at which these actin filaments meet. All right, so I'm gonna go through cross-bridge cycling a little bit faster because we did spend a decent amount of time on it last week. But the cross-bridge cycling that you see here in this figure, again, this is what is actually generating the tension, the contraction of the muscle, and can lead to the shortening of the muscle itself. And I always prefer starting down here at the bottom of this figure. So we see the myosin head right here, and then we've got all of these actins up here. So we see ATP has attached itself to the myosin head. And that ATP, remember, that's our little energy unit or currency, energy currency. So this ATP has energy. And when it moves to this portion of the figure, what's happened is that ATP has been hydrolyzed. So ATP has become ADP. And when that happens, ATP has given its energy. That energy is used to move this myosin head over to the right-hand side in this figure. And it really is like cocking a gun. So if you want to cock that trigger back, that requires energy. I've never shot a gun before. That's probably why I'm holding my hand like this. But I've seen the movies. Come on, guys. So you use your thumb to cock that trigger back, then that requires energy. So the energy that our muscles require is being used at this point right here. And I'm stressing this because at this point, we haven't generated any tension at all. We've put in energy in order to ready our muscles to produce contraction or tension, but we actually haven't generated anything yet. When we get to this top figure here, we see that myosin and actin are now bound together. And this then is where we are generating the tension, because at this point, once myosin and actin are bound together, the myosin head will return to its resting position. So in this figure, over towards the left-hand side. And when the myosin head returns to its resting position, it also moves the actin filament towards the left-hand side. All right. Are there questions on the cross-bridge cycling before we continue on? All right, good. Okay, so we did talk about a little bit about voluntary muscle control last week. We did talk about the fact that your skeletal muscles are the only muscles that you are controlling voluntarily, right? Your smooth muscle, your cardiac muscle, that's not under conscious control. And so for our skeletal muscles, we know that we're going to have an integration between the nervous system and the muscle itself. Now, I said last week, we're not going to go into neurons quite yet. I want to save that for topic 5. So we really only looked at neurons with respect to this image here, which is shown here in this slide as well. So we know that we have a signal coming in from the nervous system, right? I'm deciding I want to, flex my bicep and lift something maybe. And that motor plan, that's all happening as part of the nervous system. And we'll get to that in topic 5. So at this point, we're looking at the integration of the nervous system with the muscle in what we call the neuromuscular junction, or the junction between the nervous system and the muscle. So what we see in yellow here is the portion of the neuron that we're looking at here. This is the axon terminal of the motor neuron. And this is what is delivering the signal, hey, it's time to contract. Down here, we see we have the muscle cell or muscle fiber, and specifically we have that cell membrane. And because it's a muscle cell, it gets that fancy name, sarcolemma. Now, when the cell, the motor neuron here, releases the signal, hey, it's time to contract, what it's really doing is it's releasing a neurotransmitter called acetylcholine. So we see here in the axon terminal, these are little storage vesicles, and the green dots are representing acetylcholine. So acetylcholine is released from the motor neuron, and we will learn more details about how that happens in topic 5. Once we've got acetylcholine here released from the motor neuron, it can bind to receptors on the muscle cells. So you see these little purple guys here are acetylcholine receptors. So these are transmembrane proteins. Well, these are transmembrane proteins on the cell membrane of the muscle cell. And when acetylcholine binds to that receptor, that triggers muscle contraction. And again, we're going to get in more detail what happens when a neurotransmitter binds to a receptor, but I want to save that for topic 5. So again, I inadvertently left out the role of calcium last week, and so I did want to make sure I talked about calcium today. So we looked a few slides ago at the triad, and the triad we said was a junction between the sarcoplasmic reticulum and the sarcolemma. And the sarcolemma, remember, is simply the cell membrane. So the sarcolemma or cell membrane is shown in this sort of lighter blue-gray color here. And you notice that it kind of comes down and protrudes down here. So this protrusion or this channel that you're seeing here, that's the T-tubule. So when they talk about the triad between the T-tubule and the sarcoplasmic reticulum, this is what it's talking about. Where's my pointer? This is what it's talking about. So again, we have the T-tubule coming down, and then we've got a sarcoplasmic reticulum over here in blue and another one over on the right-hand side as well. So what I want to point out to you here, okay, is that we've got all of these red dots, and all of those red dots are calcium. So the SR, sarcoplasmic reticulum, is regulating the calcium levels within the muscle cell itself. So right here we see that it is got a fair amount of calcium stored up. The reason calcium is important is if we look at that cross-bridge cycling. So when we looked at cross-bridge cycling, again, a couple slides ago, we were looking at how myosin and actin bind together and the role of ATP in that process, right? ATP will cock the myosin head back. But what we glossed over is the role of calcium. So calcium is also necessary for contraction to occur, right? So those proteins that we mentioned briefly, tropomyosin is a protein on actin, right? And it blocks myosin from binding to the actin. So we actually need to move that tropomyosin away. So we do that by using calcium. So calcium binds to this other protein that we mentioned earlier, troponin. And when that happens, then troponin can move tropomyosin away from the binding site. Right, so I'm going to go through that one more time. So we want the myosin head and the actin to be able to bind together, right? That's how we're going to generate tension. But it can't do that because the binding site on actin is being blocked by tropomyosin. So the workaround is we have calcium released from the sarcoplasmic reticulum, and that calcium will bind to the troponin, and the troponin then will move tropomyosin out of the way. Right? So calcium is really important because without it, we would not be able to create those cross bridges or those connections between the myosin and the actin, and therefore we would not be able to generate any tension in our muscle. All right. And these videos are on the internet, on Canvas. So now that I've covered this slide in a little bit more detail, how are we feeling on the cross-bridge cycling and sort of all the players, all the pieces?
So would the question be around the steps or the functions of the different proteins, the myosin and the troponin?
So both. I mean, if you know the steps, then you would know the function of the proteins.
Okay.
So if you know that tropomyosin is the protein that is blocking the active site on actin, then you kind of know the function of it. So I'm not sure if I'm understanding the question necessarily.
No, what I mean, will you be asking what steps comes before the other, or are you asking just the function of those proteins?
You should know the function of the proteins.
Okay.
Right. And you should know the relationship between the proteins, between troponin and tropomyosin. Okay, so even though troponin is not blocking the active site, we need it because it's the one that's going to move the protein that is blocking the active site or the binding site. You should know the steps of, really generally the steps of the neuromuscular junction. So this particular slide here would be a good one to review. Again, we're not going into super detail on the nervous system yet, so I wouldn't ask questions on specific, sodium channels, for example. That's going to come in topic 5, but you should know these general steps. You should also be familiar with the steps of cross-bridge cycling. Okay.
Will you have diagrams for us to label?
No.
Okay.
Okay. So we did spend a decent amount of time last week on this series of slides looking at tension produced in muscle. So we looked at something like this already, where we have some stimulus coming in that induces a contraction. And we talked about how we can, look at this a little bit more closely and see again that there is a short delay between receiving the stimulus and actually generating tension. And that's due to all of the steps that we've been talking about so far. So the flow of calcium in that triad, so from the sarcoplasmic reticulum. Remember, sarcoplasmic reticulum, that's our calcium storage, and we need to have enough calcium available to move that tropomyosin, right, off of the binding site. So we do see here the calcium being released. And so that is in part what's responsible for this latency or this delay in generating tension. Okay, so we did look at this slide last week. So we said that when we look at the tension or contraction that a muscle is able to generate, we see a difference between this top panel and this bottom panel. And so we see these red arrows are the stimuli coming in, and each red arrow is creating a little bit of a contraction. And if you notice, after a contraction, we do have relaxation. But if the next stimulus comes before the muscle is fully relaxed, we see that tension is being continually generated as long as that stimuli keeps coming. But we see that we still have sort of contraction, a slight relaxation in the muscle, a little bit more contraction, and then again, a slight relaxation in the muscle, and this sort of oscillation here. That's unfused tetanus. And what we see for a smooth contraction, a contraction that, I'm using to say lift my water bottle up, is a fused tetanus, which is this bottom panel. So when the stimuli comes in a little bit faster, that means that our muscle doesn't have any time to relax. Remember, these little drops in tension are because there's a little bit of relaxation happening in the muscle. But if we increase the frequency of the stimuli, we get this nice sustained contraction, this even tension that's called fused tetanus. Okay. All right, so we can generate contraction or generate tension by stimulating the muscle. But then the question is, well, how much tension are we generating? So when we think about that, there's a couple things that come into play. Number one, again, that we've already talked about, what's the frequency of stimulation? But something that we didn't talk about last week, or at least I don't remember talking about it last week, is the recruitment. So we can recruit more muscle fibers if we need more precise control of the muscle. We can also talk about it in terms of how much contraction is being generated with respect to motor units. Okay, so if we look at whether or not contraction is generating tension and generating actual movement, that's when we get into our isotonic and isometric contractions. And I do think I talked about this last week. So we looked at isotonic versus isometric. And again, the difference is whether or not the muscle is changing length and whether or not it's able to actually move the load. And so the load we can think of as a weight, as pictured here in this figure from your book. And this muscle is generating tension. If the muscle is strong enough and it generates enough tension, it can move the load. So we see that the muscle has contracted, it is shorter, and it was able to generate enough tension to lift this weight. Okay. In isometric contraction, however, we see that even though our muscle is contracting and it is generating tension, we don't see a shortening of the muscle and we don't see the load being moved. Okay, so this example here is if you're lifting, say, a heavy textbook, right? You're going to be generating a contraction, generating that tension, and at some point you're generating enough tension to lift your book. But if I try to move my desk, which is much heavier, right, I can still generate tension in my muscle, but the fact that I'm not moving the load means that my muscle is not getting shorter either. And so you guys can do this right now. Grab the edge of your table. I can feel my muscle contracting, but it's not getting any shorter and I'm not moving the load, which is the desk. So both isotonic and isometric contractions are still muscle contractions. We are still forming those cross bridges. So we still have the cross bridge cycling. We still have that interplay of the actin and myosin, those regulatory proteins, tropomyosin and troponin. Everything that we've been talking about with respect to muscle contraction is still occurring. The only difference between isotonic and isometric is the muscle getting shorter? Are we moving a load? Now, one type of isotonic contractions that we didn't really talk about is eccentric contractions. So thus far, I've been talking about isotonic contractions, but more specifically, I've been talking about concentric contractions. Your muscle generates tension such that it shortens and does work, right? It lifts a load. But one type of isotonic contraction that I didn't discuss is eccentric contractions. Okay. And in eccentric contractions, the muscle is lengthening, but still generating force. And so if you think about, hopefully you guys can see this, I'm going to lift my book up, and that is a concentric contraction, which is a type of isotonic contraction. If I want to slowly lower my book, right, my bicep is still engaged. I am still generating tension. I am still generating a contraction in my muscle, but my muscle is actually lengthening. And again, we're still talking about contraction, so we're still talking about cross-bridge cycling and all that good stuff, troponin, tropomyosin, etc.
Yeah, you use contraction and tension interchangeably. Does that mean the same thing?
So contraction is what the muscle is doing.
Okay.
And when the muscle contracts, it generates tension.
Okay.
So I might have been using it interchangeably. It's a very slight distinction.
Okay.
Because contraction is going to generate tension, and tension is being generated by contraction. So I apologize if I was using them interchangeably, but that's the distinction there.
Okay, thanks.
Okay. So I believe the rest of the slides are more or less related to energy and muscle fatigue. And I do remember I spent a decent amount of time on this last week. So I wasn't going to go into it unless you guys wanted me to. Yay or nay?
I have a great question.
Oh, yeah.
How many questions is it?
30.
To 60 or just 30?
30.
Okay,
It's 30 to 1,000. No, there's 30.
And is it like 33.33 points like last time, or?
Yeah. So
Okay.
it's gonna look very familiar to the previous exam. So there's going to be 30 questions that are worth, right, 3.33 points. And then there's going to be 3 questions that are worth 1 point. And those are the extra credit questions. Now, I did play with the settings, and I hope that it does show up, because you guys told me that you weren't aware of which 3 questions were the extra credit questions. So I did try to be a little bit more careful with how I set up the test bank, but I didn't, with the Canvas controls, it didn't seem super obvious a way to do that, but I'm hoping that it shows up. At the very least, you could look at the point value for the question. So if the question shows up and it says it's only worth 1 point, then you know that that's the multiple choice question, or sorry, that's the extra credit question. But if a question pops up and it's worth 3.33 points, then you know that's one of the actual exam questions.
So did you increase the time this time to 45 minutes?
40 minutes.
40 minutes.
Yeah.
When will you post the Unit 5 module?
Pardon?
When will you post the Unit 5 module?
This weekend. I'm working on it. So I finished up exam 4 today, and so tomorrow I'm going to start in on topic 5. So I'm like barely ahead of you guys.
No rush, just wondering.
Yeah. As far as topic 4, again, I went over most of these last week, all about the fast and slow fibers. And then we talked about the glycolytic or the anaerobic fibers versus oxidative. So I wasn't going to go into that again, unless there was a question on any of this. Okay. All right. All right.
Back to that slide where you have fast oxidative.
This one?
Yeah.
Yeah.
So for, oh, I see. For endurance is slow oxidative fibers. That question did come out in the previous.
Yes. Yeah. I should highlight that. So slow versus fast refers to the speed of contraction. So that's one distinction if you're a slow fiber or you're a fast fiber. And then the word oxidative versus the word glycolytic refers to how that fiber is generating ATP. Is it using aerobic respiration, which produces ATP through oxidative phosphorylation, which is where this word oxidative is coming from? Or does it utilize anaerobic processes, so glycolysis? And again, that was something we described back on these slides, anaerobic versus aerobic pathways to generate ATP. All right. Oh, so are there other questions on this? Okay. So in terms of what fibers we have where, we have a mixture and everybody's different. So different muscles on one person might have a different distribution or different proportion of these 3 fiber types. And then, of course, genetics is going to play a part as well. But what you need to know here, again, is what slow versus fast means, what that oxidative versus glycolytic means, and what these fibers are good for. So again, a slow oxidative fiber is good for low intensity endurance movements, right? These things aren't fast movements, so we don't need super fast contraction, but we do need it to be able to contract for long periods of time. So it's good that it's able to utilize aerobic respiration to generate ATP. So if you break what these words mean down, it does make sense in terms of the types of movement that these fibers are responsible for, right? So high intensity movements that we don't need to do for very long can utilize fast glycolytic fibers. I don't think we covered effects of exercise last week. But exercise, of course, is important, and hopefully we're all still getting a little bit of exercise, even though we're all on lockdown. But exercise causes a lot of changes, and I think a lot of us think about muscles getting bigger, and that, of course, is happening, right? If you're, lifting a bunch of muscles, sure, your muscles are going to get bigger. But What also happens with exercise are these three things here. So we get more capillaries into the muscle. So we're getting more blood supply coming in. Our muscle cells can increase the number of mitochondria. And mitochondria, remember, that's where respiration is occurring. That's where our cells are able to generate ATP. So if your muscles, you're exercising your muscles and you need more energy, then your muscle cells say, oh, all right, well, we better have more mitochondria so that we can have enough ATP available to, complete the exercise. And then the last is that we see more myoglobin or increased myoglobin as a result of exercise. Now, I don't remember if I got into smooth muscle last week.
For the capillaries, you said increased blood flow and the mitochondria generates energy. For myoglobin, that is for what?
Oh, sorry, the myoglobin is what's binding oxygen. And so that's important because the aerobic respiration requires oxygen.
Okay.
Right, so increased capillaries is going to bring more blood to the area, and the blood is carrying the oxygen. But the myoglobin is what's binding the oxygen in muscles. So when you look at smooth muscle, I like to compare it to skeletal muscle and how it's different. And we did talk about the fact that smooth muscle is not under voluntary control. So that's a huge difference between smooth and skeletal muscle. But there's a lot of other differences. So skeletal muscle has all those striations, those bands that we saw in those figures, but smooth muscle fibers don't have those striations, which is fine. So if we look at this figure here, right, this yellow, all of this yellow, that is actually your nervous system coming in. So we still have a connection between the nervous system and the muscle, but again, the difference is that this portion of our nervous system is not under conscious control. So this is part of our autonomic nervous system. And again, we're not going to go too far into this until we get to topic 5. I'm just pointing out the similarities between smooth and skeletal muscle in that we do have that junction or interplay between the nervous system and the muscle tissue. It's just a little bit different. So if we look at this figure here on the left, we see some terms that we're more or less used to. So here we're looking at part of the muscle cell, and just like we learned with skeletal muscle, smooth muscle has a sarcoplasmic reticulum that is responsible for regulating calcium levels. So again, we see plenty of calcium here as well. But it's a little bit different because we don't see those triads. We don't have the T-tubules. We don't have so much of the SR isn't as developed as it is in skeletal muscle. So it doesn't have that triad structure that we saw a few slides ago. But a few things, in terms of contraction is we are still getting contraction generated. It is still relying on calcium being around, ATP being around. So there are some similarities between smooth muscle contraction and skeletal muscle contraction. A couple important differences when we're looking at smooth muscle contraction is the fact that the cells are actually coupled together with gap junctions. And gap junctions are a type of intercellular junction, so a junction between two neighboring cells. And we looked at this in topic 1, I think. So we looked at tight junctions, gap junctions, and anchoring junctions. So gap junctions are connections between two cells that actually physically connect the cytoplasm of one cell and the cytoplasm of the neighboring cell. And this is important because essentially if the inside of cell A and the inside of cell B are connected because of this gap junction between them, then that allows for coordination of those cell activities. So the coordination is coming because these cells are actually connected with gap junctions. And that enables that nice synchronous contractions in smooth muscle. Smooth muscle is slower and requires less energy than skeletal muscle. I think that more or less makes sense based on the type of function that smooth muscle is carrying out versus skeletal muscle. But again, we do have some similarities: actin, myosin, ATP, calcium, all that good stuff. As far as regulation of smooth muscle contraction, we're not going to get here yet. This is, again, what we're saving for topic 5 when we get a little bit more with the nervous system. Oops. All right, and then the very last slide, muscle development. I think I might have talked about this briefly last week. But when we're talking about development of muscle or embryonic development of muscle, again, we're not an embryonic development class, so we're looking at just a really cursory overview of embryonic development. So with the embryonic development, we've looked at the 3 layers: the endoderm, mesoderm, ectoderm. The mesoderm is what is giving rise to our muscle tissue. So here we see those mesoderm cells differentiate into myoblasts, and then myoblasts can divide and actually then produce all of our muscle cells. All right, so are there questions? Should I go back to anything? What do you guys got for me? What questions?
You have 5 extra credit points. Is that a question?
No, but you can have 3 extra credit points. 3 extra credit points on the exam tomorrow. Or I should stop saying tomorrow, because you can take it anytime this weekend. Okay. So that is the sort of quick and dirty review of topic 4. If you guys don't have any more questions, I can go ahead and let you guys go so that you can carry on with whatever you guys have going at home tonight. And just make sure you take that exam sometime before Sunday at 11:59 p.m. Right, I will be adding stuff to Canvas for topic 5 throughout the weekend. My slides should be already up there, but I'll be adding some more videos and some extra resources. And I don't know if I'm going to get the homework assignment posted yet this weekend. If not this weekend, then like Monday, Tuesday next week. Okay. Are there other questions?
When will the assignment be due? When are they due?
I usually give you guys a week, so it depends when I post it.
Okay.
But it'll likely be due Sunday the 26th at 11:59 p.m.
Okay.
Okay. And it's not going to be like a big thing. It's not going to be a big thing, so don't worry. All right.
What's
if you have more
the sorry, what's the homework gonna be? Like what's the best how are we gonna get like max points on it? Basically, what are you looking for?
So my homeworks are fairly simple. I actually haven't decided on what the homework for topic 5 is going to be yet, because I just have not even started topic 5 really. I've been kind of obsessed with topic 4, so I don't know what the homework assignment is going to entail. I will try to be clear with the instructions on the homework assignment in terms of what I expect and how to complete the homework assignment. But if anything's unclear, please just message me and let me know, and I can clarify the instructions for you. Okay. So homework should be a tool to help you learn the material. It shouldn't be busy work. A good homework assignment, I should say.
Sounds good.
All right, ladies. So thank you for joining me today on Zoom. I did record this, so hopefully I can figure out how to get this up on YouTube. So if you want to go back and listen to anything, I actually think I should be able to get this up tonight. Okay. So everybody have a great week. Good luck on the exam. And I will see you ladies next Thursday. All right.
Okay. Thank you.
Take care.
Bye.
Bye.